Hello, neuro friends. So, this week we're going to be investigating neural circuitry by using the Brain Explorer software that's provided by the Allen Brain Institute. So, I'm going to start off by giving a brief tutorial on how you can use this software to investigate the neural circuitry for the regions that you're interested in. So, from looking over the protocol here, first we're going to have to download the software in order to use it. And so to do this, just simply click on the link that's provided in the protocol, and that will take us to the site for the Allen Brain Institute. Then click on the link for Brain Explorer, and you'll see that there's options for downloading on either Windows or Mac. And so I'm going to go ahead and do the Macintosh download since that's what I'm working on right now. So returning to the protocol, the next step after we have our software downloaded is to install the different atlases for the software. So as soon as you open up the program, you'll get a message like this asking you to install. Go ahead and just install all of the different atlases on the bottom here, the Allen Mouse Brain Atlas, the Developing Mouse Brain Atlas, and the Allen Human Brain Atlas as well. Okay, so now that we've got everything downloaded and installed, we're going to go ahead and find our region of interest using the software. So to do this, we're going to go back to the Allen Brain Institute website, and we'll put in our region of interest into the search. So for example, I might be interested in substantia nigra, and that will pop up. Go ahead and close. We can close some of the default regions that show up as well, like the retina. And then we'll see a whole list of experiments that show up. I'm just going to go ahead and click on one of the experiments here in the list. And you'll see you'll get a couple of different views of the projection of the tracer, the location of the tracers back in the midbrain brainstem where we'd expect. And then it's spanning out to all these different areas of the brain, which you can see the tracers labeled with this fluorescent green label on it. We'll also get a picture of some coronal sections of the mouse brain as well as we look at this. And so you can see if we zoom out a little bit, we have a coronal section that's pretty far posterior into the brainstem. And here is the location of the injection site, which if we pull up actually some of the Brain Atlas pictures that we looked at before, we'll see that this site is right where we would want it to be, back in the midbrain brainstem where the substantia nigra is located. So we found our region of interest at this point using the online website, and now we're actually going to initiate the software to get a more in-depth look at some of the neural circuitry. So we'll go back to the web page where we were, and you'll see that there's actually a tab or a button on the very far top right, this 3D box, which is going to open up the Brain Explorer software. So go ahead and click on this box. And what you're going to actually open up is a map of all of the connections that span out from your region of interest. And so you can see this spot of dark red squares, and this is where the tracer was actually taken up by the cells. You can then use the right click to move around in any direction that you want to see different angles of this tracer. You can rotate it. What we're actually going to want to do though is that we're going to want to look, even though there's this extensive connectivity everywhere, one helpful thing to do to pinpoint a connection is to look at the strongest connections and where most of the connectivity is going to. And so you can do this by adjusting this box here and adjusting it so that you're getting only to the most intense projections and to the most dense projections as well. I'm going to go ahead and click on one of the projections here with a left click. And you'll see that this is actually showing the caudate putamen area, which we know is actually part of the basal ganglia system. And if we go back to the lecture slides, we'll see actually that there is this strong connectivity between the substantia nigra and the caudate putamen, which makes a lot of sense. If we cut back to these lecture slides, we'll see again, here is the basal ganglia structures, the caudate putamen, what we just found the substantia nigra is connected to. And remember, this is part of the basal ganglia system that's part of motor movement. So if we look at this image here, you have the basal ganglia that's embedded in the frontal cortex or beneath the cortical areas of the frontal cortex here. And it has, again, this strong connectivity to the substantia nigra, just like we traced out from this actual mouse brain tracing system. And so if we go back to our mouse brain atlas, we'll see again, here's the structure, the caudate putamen that we traced out. What I'm going to go ahead and do is press Command E, and that's going to show that region there of interest that we just mapped out. And I'm also going to make this transparent by going to the atlas function and clicking transparent. So you can see a little bit more clear view underneath the structure here and see our tracer projection. Now let's explore some of the other potential connections that are here. So the next connection that pops up actually is, if we click on another connection, is we'll see actually there's a connection to the thalamus. This may not be so surprising because we know that the thalamus is a relay sensor, or a relay for all sensory information, including touch information that may be important for motor movement. So if we go back to that lecture again, here we are where we initially introduced the thalamus as part of the course. And remember, the thalamus is part of the between the brainstem and the forebrain. And again, it's a sensory relay center. So if we go back to our Brain Explorer software again, we'll see, again, we'll click Command E, and that will show up as our next region, the thalamus, which is right in between the brainstem where our substantia nigra is located and the caudate putamen that's in the forebrain. We can go ahead and rotate this around a little bit more to kind of give you a more in-depth look. If you hold the right click and actually spin your mouse, you can actually start rotating the brain. You can even add on more to the brain. If you keep clicking Control E, it will add on more and more of the connectivity and the structures as you go along to get a very in-depth look of the entire brain here. So go ahead and rotate this back around to where we started. Keep adding on more and more brain, and eventually you have an entire brain with a nice transparent view. And so there we have it. There's a couple of structures that we just mapped out from substantia nigra. Hopefully you'll be able to do the same thing with your projects. I showed you a couple examples. One connection that we had actually talked about in class, the substantia nigra to caudate putamen connection, and an additional connection from the substantia nigra to the thalamus that we haven't investigated, but you could go ahead and investigate with your own research.